So good afternoon, everyone. Thanks for dialing in. My name's Mark Shackleton. I'm the clinical lead of the MPCCC's Precision Cancer Program and also the Director of Oncology at Alfred Health. I'd like to open today's seminar by acknowledging the traditional owners of the lands on which we join from today, and respectfully acknowledge their elders, past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community gathered amongst us this afternoon. The MPCCC Precision Cancer Program is a community of researchers and health professionals and academics from across Victoria that have an interest and a shared interest in precision cancer medicine. The program has been running for must be at least 3 years or so now, and working to establish better access to molecular sequencing for cancer patients across Southeast Victoria, or Southeast Australia really, as well as particularly Victoria, and also to provide fora in which the results of such sequencing can be interpreted to inform patient management. As part of that, we host several tumour-specific molecular tumour boards, and please contact MPCCC administration to find out more about participating in those. They're certainly these seminars are certainly a very important part of our program, and have been a very successful forum for us to collect together to hear about some of the latest research going across the MPCCC partnership, but also more widely across our country and even internationally. Today, I'm very pleased to welcome Dr. Malaka Ameratunga as our guest speaker. Mal is a medical oncologist and an early career researcher with an interest and very impressive academic pedigree in early phase clinical trials and in drug development. Mal, following a very successful period of fellowship work particularly at the Marsden Hospital in London, Mal is a relatively recent recruit to the wider kind of Monash Uni affiliate precinct, doing both clinical work as well as currently finishing off a PhD in the Central Clinical School at Monash University. He's got a lot of really interesting ideas and insights into opportunities for improved cancer drug development. And his topic today is Beyond Driver Mutations, using molecular profiling to inform drug repurposing in the clinic. We look forward to your talk, Mal. There will be some questions, some minutes for questions at the end of the presentation, but for now, everyone could just turn off their microphones and videos, and I'll hand over to Mal. Thanks, Mal.
Thanks, Mark, for the introduction. So yes, the topic of my talk today is Beyond Driver Mutations, using molecular profiling to inform drug repurposing in the clinic. This first slide is really just a historical perspective on the molecular error in solid tumours. So the proof of principle of targeting tyrosine kinases was really established in CML, in hematologic malignancy, with imatinib showing efficacy against the BCR-ABL fusion oncoprotein. The story in solid tumours is a bit more circuitous. So gefitinib was actually developed in 2003 for EGFR overexpressed tumours, and actually they were really interested in squamous tumours, which turn out to be the type of tumours that don't respond to gefitinib. And in 2005, some astute clinicians observed that the patients that were on this treatment that were responding were all female non-smokers and Asians, and put two and two together and worked out that actually they had mutations in the kinase domain of the epidermal growth factor receptor, which conferred sensitivity to this drug. And with imatinib, that necessitated the development of cytogenetics testing so that we could get patients treatment. And gefitinib necessitated the development of targeted panels so that we could actually identify mutations to get our patients these treatments. And that really kick-started the era of the molecular error in oncology. And the first phase was with a lot of multi-targeted dirty tyrosine kinase inhibitors such as sorafenib, sunitinib, and pazopanib. They were targeted tumor types, but not at specific mutations. There were also sort of less successful but active drugs like lapatinib for HER2, which targeted overexpression protein by ISH. And then in 2011, the next sort of wave of drugs came through. So crizotinib in ALK, vandetanib in RET, and vemurafenib in 2011 were published. And those, along with a small molecule but not a kinase inhibitor, olaparib for patients with DNA repair defects, palbociclib for breast tumors, sonidegib for BCCs, led to the development of tumor-specific panels. So if you have a lung mutation, you get a panel looking for ALK. If you've got a gynecological cancer, you get a panel looking for DNA damage defects. And then the last few years has been another explosion, and a number of these therapies are active and FDA approved, like larotrectinib in NTRK, metinib in MET, alpesolib for PIK3CA, erdafitinib for FGFR, and sotorasib for KRAS. And many of these are actually not available in Australia at the moment, despite their demonstrable activity. And so, this is sort of propelling the need for comprehensive sequencing. The other thing to note, and I put it here in red, is that the first pan-cancer FDA approval came in 2017 for an immunotherapy, so not really the scope of this presentation, pembrolizumab, but the pan-cancer approval of drugs obviously opens up a whole other slew of options for patient treatment, which may change the way we do things in the future. So where does that leave precision medicine in the clinic in Australia in 2022? There's actually a diverse range of sequencing panels available, and they all have different number of genes on them, different out-of-pocket costs, different timelines. Unfortunately, tumor-agnostic comprehensive sequencing is not yet standard of care. And the comprehensive sequencing that we have, the availability is limited by cost, time, and tissue availability as well. Which leads to a bit of the background, the talk. And so, despite this explosion of active drugs, we still have limited options for poor prognosis tumors. And I'm talking about tumors here for which there might be one line of chemotherapy, rarer tumors, not the ones with these known biomarkers, or the breast and lung cancers where they've got 3 or 4 available lines of treatment. And part of that's because we've got an inflexible regulatory and treatment milieu, which I'm going to talk about, and also the lack of incremental progress in this patient population. And a lot of this is to do with the patient journey that we have in this group of patients. So if you've got a patient with an aggressive cancer, often, if there's a trial, great, we'll put them on a trial. But if not, the licensed therapy, and what we're talking about here is usually chemotherapy. You try it, and if you've got the resources and capability of getting sequencing, either a targeted panel or a comprehensive panel, you do that until there's progression. And then at this stage, you come back and there's often, no further options. If you have sequencing options which open up more licensed therapies or trials, you go down that path. And if there's no trials, but a sequencing result which would be interesting, there's also an issue of funding and getting funding for those drugs. So it's not even straightforward, even if there is a licensed therapy available, if there's no funding for it. If there's no sequencing results available and you can't get sequencing done quickly, or if there's no funding, we're looking at best supportive care, which might be a second-line chemotherapy, or best supportive care or the best available treatment, which might be a second-line chemotherapy. And because these patients don't get novel agents, we're not learning. So there's been no, a lack of clinical learning in this subgroup of patients. So, if these patients are lucky enough to get onto trials, often they'd be considered for phase 1 or early phase clinical trials. And these trials are predominantly designed to find the right dose of a drug. So their endpoint is that of safety first, and efficacy comes as a secondary endpoint. Because of the rigorous focus on safety, they typically exclude any patients that have comorbidity, which functionally excludes any patients with real-world aggressive cancers. And as I mentioned before, the logistics around sequencing and precision oncology is a little bit ad hoc still. It's not optimized for time. Consequently, if you've got a patient with an aggressive cancer where sequencing may take some time, you might end up missing out on a comprehensive sequencing panel. So in terms of these trials, a standard set of exclusion criteria in a phase 1 trial would be the presence of any brain metastases, any other uncontrolled medical condition in addition to the cancer, kidney dysfunction, liver dysfunction, any second cancer. We're increasingly seeing patients with multiple cancers, a life expectancy less than 90 days is an exclusion, and then a performance status greater than 1, which means patients that aren't really fit and well would be excluded under that category. And then moving forward, there's increasingly additional criteria such as nutritional criteria which are coming in, such as albumin greater than 30, which excludes even more patients in this group. And even if you pass all of these criteria, there are additional hoops, such as being 4 weeks since the last treatment, and then availability of the slot. So for instance, if you had 5 patients that you wanted to put on one trial, you wouldn't be able to treat them all at the same time. You'd have to wait. So all of this, creates problems in this group of patients. And we are working on solutions. So the 2 probably best noted clinical trial designs for these sorts of problems are basket trials, illustrated on the left here, and umbrella trials. So a basket trial is if you're looking at a biomarker. So say BRAF mutation, you've got patients. So BRAF inhibitors are licensed really in melanoma, but if you have another tumor type with a BRAF mutation that would confer sensitivity to a BRAF inhibitor of any tumor type, you'd go into this basket study testing that biomarker. And umbrella trials are a bit more complicated. So here you start with the tumor, and then you have a number of sub-studies opened under that umbrella, depending on what so once the sequencing occurs, you've got biomarker A, you go on trial A, B, you go trial B, C, you go trial C, biomarker D, trial D, no biomarker, you go into arm E of the trial. But these still don't solve the problems of sequencing and availability. They're also resource intensive, and often centralized sequencing is slow. And the feedback I've received, certainly anecdotally at least, from my patients is that a more inclusive framework would work better for our patients. And the other thing I should sort of say is that actually the response rates of phase 1 trials are actually poor. So the overall response rates are 5% for first-in-human studies. In one publication, they're reported as high as 15% in biomarker-enriched trials. But even an updated meta-analysis published last year in the molecular error alone, so only in the last 4 or 5 years, had 5% response rates in an unenriched population and 12% in a single tumor type biomarker-enriched population. And the real response rates are actually lower than that because there's publication bias to positive results. So if you've got a negative study, it takes a long time to end up being published. And then there's also the fact that there are many second-in-class medications that classify as phase 1 trials. So for instance, there's an approval for olaparib, a PARP inhibitor for prostate cancer. Then drug company B, C, D all have PARP inhibitors that are undergoing phase 1 trials, and a priori, they have got a very high chance of having a response rate that's similar to olaparib, given that it's a class effect. So those will also skew the results of these response rates positively. So overall, actually, even the best trial option that we have is often not very good. And I guess an alternative paradigm that I have been thinking about for a while is that, well, what if instead we had this sort of patient, so typically a patient that would be otherwise considered for a phase 1 trial. You consent the patient, you pass the QC and then sequence the patient very quickly, and do a molecular tumor board assessment quickly. If you've got a validated target or a licensed therapy or a trial that's available, offer that treatment subject to the funding. But if you don't, could we actually do a computational analysis to work out existing off-the-shelf medications that might actually work in this patient population? So therapy for which we know the dose, the schedule and the dose that's previously undergone a phase 1 trial. Treat the patients and then learn from the patients that respond or don't respond with more formal trials or formal preclinical research of the more interesting responses. That brings me to the repurposing platforms. And so the platform that I'm familiar with is called ProbeMiner. It was developed at the Institute of Cancer Research by some of my collaborators there. And it wasn't designed as a repurposing platform. It was actually designed as due to problems in the medicinal chemistry literature regarding the accuracy of chemical probe data for ligand receptor interactions. And it's curated from large public databases, especially the Cancer Knowledge Base. And they took six domains, so potency, selectivity, cell potency, structure activity, relationship, inactive analog and pains, quantifiably ranked them, heavily weighted them to potency and selectivity, which are likely to be the most important, and then come up with a rank for for probes for that specific target. And in addition to this, to ProbeMiner, we've also collected data from other repurposing or trial allocation platforms, including the Broad Repurposing Hub and Topograph, which is what the Garvan uses to allocate people to the most clinical trial. So this is the type of data that you'll get from ProbeMiner. So if you've put in the gene target, which is BRAF, this is a list of the top 20 chemical probes for that target. If you've got a name that's cancer, etc., they're chemical probes that are not licensed drugs, but there are also licensed drugs in here, including vemurafenib, encorafenib, and dabrafenib, which are in fact the 3 licensed drugs for BRAF for BRAF. So it shows the validity of this platform and the type of results that you get. And then this is dabrafenib selected here, its chemical structure and how it was weighted on the scoring system. So there's still some problems with this. So the frequency and therefore cost of clinically actionable repurposing events is really unknown. There's a major consistent problem anyway in interpretation of next generation sequencing reports with the annotation of unknown variants. And there's a problem that in silico modeling is based on the chemical ligand interaction of these curated databases, but it doesn't give any functional information. So for instance, that platform tells us that dabrafenib binds is likely to bind BRAF, but it doesn't tell you that inhibiting BRAF actually causes tumor cell death and therefore responses in patients. So it doesn't give any functional information. So the aim of this project was to really test the hypothesis that drug repurposing is viable for this subset of patients, and to demonstrate a feasibility of this a repurposing approach. And so the method we used was to take patients sequenced on the most clinical trial through the MPCCC. And there were 94 patients with NGS panels through the MPCCC, which were performed. The American College of Pathologists have developed this tiering system for annotation of variants. So, tier 1 is sort of FDA approved. Tier 2 is things that we think are going to work, but we don't know it yet. Tier 3 is unknown, and tier 4 is known and they're benign. But that's still actually quite subjective, and the reports changed over time. So as a consequence, we didn't use this classification scheme for curating the mutations, but did it manually with 2 oncologists. And the reason for this is we wanted to identify, mutations that you wouldn't have a standard therapy for, to exclude that from our analysis. And so this is the panel for the most clinical trial. So the Illumina TrueSight Oncology TS0500 panel was used for the vast majority of patients. So that's 523 genes, and these are the genes listed for single nucleotide variants and indels, 55 genes for fusions and splice variants, microsatellite instability, and a tumor mutational burden, and a small number of foundation panel, which is 324 genes, but also MSI and TMB. In terms of variant classification, so we really focused on gain-of-function mutations, and the reason for doing this is it's much more straightforward to do. So obviously, if you've got a loss-of-function mutation, you need to find a drug that's a synthetically lethal combination. So targets a different gene to the lost gene. Whereas if you've got a gain of function, you can actually directly target the gene that's altered. And so the way we classified mutations was first look at the literature. So if you've got something like this, BRAF, a V600E, this is clearly a hotspot. So there's 55,000 reported cases in COSMIC at this hotspot. It's a pathogenic variant. We classified truncating mutations as loss of function. We classified amplifications as gain of function mutations. And then if you had non-synonymous single nucleotide variants, we did it case by case. So if it was a kinase and a hotspot domain mutation, it was possibly labeled as annotated as a gain of function mutation. And specific mutations that have previously been orthogonally functionally validated were also annotated as gain of function mutations. So here we've got what would be classified as a variant of unknown significance, PIK3C2G R1034H, which is identified in one of these reports. This is a PIK3C2G C2G gene in COSMIC, and as you can see at 1034, there's 4 mutations. There's no hotspots. In fact, there's the maximum is 18 previously described mutations. So this is a very relatively uncharacterized gene. The mutation results in an arginine to histidine substitution, which is known to alter pH sensitivity, which tends to increase responsiveness to the ligand, and it's in the kinase domain. And that signature is also associated with pathogenicity across cancer. And finally, using computational prediction, these aren't 100% accurate, but on the FATMM score, which is what we used, the pathogenic score was 0.99. So this was annotated as a possible pathogenic gain of function mutation. So then once we got these gain of function mutations, we ran them through our repurposing platforms, grouped the repurposing events into various categories, and then tried to identify the better the more novel repurposing events. And we used topograph from the Gov. So a most report that has an abnormality, they link it through the database topograph to find, to link it to clinical trials. So any abnormality that would be automatically flagged to a clinical trial, we excluded from this analysis because we thought that would be equivalent to being a tier 2 mutation. So this is the results of the study. So there were 94 patients. The median age was 63, and a third were elderly, so greater than 65. The main thing to point out from this slide is that actually these are not what we see in the clinic routinely. So there's hardly any breast tumors, hardly any melanoma, relatively fewer lung and prostate. There's overrepresentation of tumor types for which there's less available therapies, including upper GI, hepatobiliary, and pancreas, and lower GI, as well as gynecological tumors, and other is overrepresented. In terms of histology, adenocarcinoma is traditionally thought to have more likely to have driver mutations, and that was only about 2/3, and a third were other, including poorly differentiated carcinomas and others of greater than 20%. And these patients are mostly unlikely to respond to immunotherapy. They didn't have microsatellite instability, and only a small proportion had high tumor mutational burden. So this is a poor prognosis group of patients. And this is just that data represented visually, but of note, in the other tumor subtype, half were unknown, and another 10% were cancer of unknown primaries, and then a smattering of peritoneal, anal, and thyroid cancers. In terms of histology, the other histology subtype were serous adenocarcinomas, mucinous adenocarcinomas, glioblastoma, papillary cancer, and small cell cancer. So all of these, again, cancers for which there aren't a great deal of therapeutic options in general, which makes sense because they were referred to this for most for that purpose. And so this is a breakdown of the mutations that were identified. So there was 176 gain of function mutations that were identified. 104 were amplifications, 65 non-synonymous single nucleotide variants, and then a small number of fusions and rearrangements. Although prominent oncogenes, including KRAS, TP53, BRAF, and EGFR are represented, there are also a lot of less prominent genes that are shown here, including 64 and other for amplifications and 22 non-synonymous other mutations. Even for the genes that were mutated, common oncogenes, it wasn't always the most common non-synonymous single nucleotide variant. So, for instance, in BRAF, V600E is the most common mutation, but 3 out of the 4 were non-canonical BRAF mutations out of the 4 BRAF mutations that there were. Similarly, these, some of these are less common TP53 mutations, and certainly some less common KRAS mutations as well, although here at least G12D was the most common in the study. At a drug level, and this is for all patients, there wasn't much overlap between the drug repurposing events across the 3 platforms. So on Topograph, there were 178, ProbeMine 195, and the broad 127, and then very small regions of overlap. There were 43 new drug events for which there weren't active clinical trials or available licensed therapies that are currently studying those genes, locally at least. Those 43 drug events were across 19 genes. So some of those genes had more than one drug. And then this is the overlap diagrams at a gene level. And again, just showing that there isn't actually that much overlap between these repurposing platforms. A good thing that there is an overlap with Topograph, which is the allocation for trials. I don't know as much about the Broad Institute methodology, but certainly different results to ProbeMiner. In terms of the actual mutations that were identified, so non-synonymous single nucleotide variants, we've got 35 here, 34 amplifications here. And the key thing to note is that, there's a lot of different genes that only came up once. And this is what you see in real life. So, a lot of genes that are rarely impacted, because they're rare, there aren't studies that are evaluating them because it doesn't, it's not commercially viable, etc. And although there was still the most common genes were still there as well, which validates that these patients are still representative of the tumor types that we see. And this is a consort diagram of the results. So there are 176 gain-of-function mutations. We removed 48 that have FDA-approved therapies, leaving us with 128. 16 were being evaluated on trials through Topograph, leaving us with 112. 95 didn't have any, useful probes on Probe Miner, which left us with 17 mutations, of which 2 were essentially duplicates. And these are the 15 mutations that were left. 10 mutations were removed that, are not completely novel, and 5 we felt were quite novel. And this is a little bit subjective, last part. So I guess the results of this data is that there were quite a significant number of novel events. So 5 out of 94 that were quite novel, unexpected. Certainly, practicing oncologists wouldn't be thinking about those. 15% of patients would have had a repurposable drug, at least theoretically, on this analysis. And the thing I'd like to point out is that actually only 11% of the kinomes actually currently characterized and included. This is definitely going to improve over time. And actually, the licensed number of drugs are increasing steadily year on year. So, certainly interesting results. What about functional data? And can we actually show any functional data to show that this approach might work? So there are publicly available functional data sets that can be interrogated, and so that the most straightforward to use is DepMap, which is publicly available data from the Broad Institute and Sanger, with harmonization across the Cancer Cell Line Encyclopedia. And you can actually use these data sets to interrogate combined gene expression, CRISPR viability, mutations, RNA interference, and combine that with drug sensitivity studies. There is complexity in analyzing the data because there's a diversity of cell lines, so it's all tumor types. There's cell context dependency as well. So for instance, in EGFR, which we talked about, the epidermal growth factor receptor in lung, we know that EGFR inhibition works in lung, and it doesn't really work in the same way, at least in colorectal cancers, because they're wired differently. And obviously that sort of information isn't really captured. And we know that after sort of years and actually over a decade of study of that topic, that sort of information isn't captured in this interrogation. So I've included this slide to sort of to show you what the data looks like. So this is a known biomarker drug combination, this slide. So this is looking at the combination of the epidermal growth factor receptor with the small molecule tyrosine kinase inhibitor erlotinib, which is licensed, has a very high response rate in EGFR mutant lung cancer, and doesn't really work in much else. So this is what the data looks like in this sort of example. So I've got 2 different types of plots here. And the left one is an expression plot, and the right is a CRISPR dependency plot. The y-axis will change depending on the data set that was available. But the point to make is each dot represents a different cell line. So as you move down on the y-axis, you're getting more sensitivity to the drug. The x-axis is the opposite. So for expression, as you go across on the x-axis for expression, you're getting increased expression of the biomarker EGFR. The x-axis for CRISPR, it's negative. So as you move leftwards on the x-axis, you're more dependent on EGFR on a CRISPR screen. So what you can see is that there's I've also included a simple linear regression curve from the scatter plot, and theoretically, the steeper the gradient on that curve, the stronger or the better the relationship between the biomarker and the drug. And what you can see is the expression has got a weak relationship, but there is this population of patients that have high EGFR expression and sensitivity to erlotinib. a stronger relationship on the CRISPR side, and again, a population of cell lines that have an EGFR dependency and are responsive to alatinib. So that's a really clean EGFR alatinib, and you see how tricky the data is to interpret in that context. This is another one, another clean biomarker drug combination. So BRAF mutations and dabrafenib. Again, dabrafenib works in most tumor types that have a BRAF mutation, except for maybe colorectal cancer. And here you can see, a weak relationship with expression, a stronger relationship with the CRISPR dependency, and populations of patients that have high BRAF expression or BRAF dependency that are very, very sensitive to the drug. Now, interpreting this functional data with repurposing is even more complicated, because most of the repurposed drugs are not highly targeted because they're actually targeting more than one target. And secondly, because we're using licensed drugs, they're already licensed for another indication. So some of the negative controls on those graphs will actually be, the original indication for the for that drug. And then CRISPR dependence is tricky because it's difficult to interpret if the mutation of the gene is not well characterized. We know about these CRISPR dependencies for EGFR and BRAF from again, decades of work on them. But we can't really identify a CRISPR dependence a priori, at least not with this method, if it's a novel mutation. And so this is an example of a licensed biomarker repurposed drug. So imatinib was developed for CML originally, and it also works in KIT mutant melanoma and KIT mutant GIST tumors. So, and we know this is used in the clinic. It's got a high response rate. And what you can see here is that, you can't, it's very, very difficult to see any sort of relationship between expression or CRISPR dependency and activity of the drug. So with that in mind, moving on to the data from the drugs and targets that we got. So this is a subjective classification, but there was these drugs here I'd characterize as, really quite unexpected, with, some promising or interesting functional data. This second column is things that are unsurprising, but actually, potentially actionable, with, again, some interesting functional data. And this here is completely novel, but I couldn't find any supporting functional data. The ones that I've indicated with the star were mutations, and most of the others were amplifications. So, Indomethacin, the androgen receptor, and I'll go through, some of the others of these, this data in the next few slides. So this is one of the novel mutations that we had. So PIK3C2G, a previously, a variant of uncertain significance. What you can see is a weak relationship. Midostaurin is a multi-targeted tyrosine kinase inhibitor used in hematological malignancies. It's very active against a broad range of targets. So makes it, there's going to be some degree of activity in most of these cell lines, but what you can see is some, modest activity in cells that have high PIK3C2G expression. And because this is a mutation, actually, the CRISPR data might be, more relevant than expression data. And you can also see a relationship between the dependence on PIK3C2G and the sensitivity to midostaurin. So that's, some interesting data on that. This is another one. So FLT4 was again a mutation rather than an amplification. A very weak relationship between expression and sensitivity to tivozanib, but there was this population of patients that had high expression and were quite sensitive to tivozanib, and again a weak relationship with CRISPR as well, but a proportion of cell lines here that dependent on FLT4 and quite sensitive to tivozanib. Cytarabine is very interesting in the sense this is actually cytotoxic chemotherapy that came up on a repurposing screen, and again, because it's cytotoxic, it's always got some degree of activity, and again, modest relationships and a proportion of cell lines for which there's high expression and or dependency and significant activity for cytarabine. And probably this is probably one of the this would be our ideal type of drug. So indomethacin is a non-steroidal anti-inflammatory drug. It's quite routinely used. It's very, very well tolerated. It's actually it's almost an over-the-counter drug. And that came up as actually the #1 hit for binding the androgen receptor over natural ligands such as testosterone and above many licensed therapeutics that target the androgen receptor. Unfortunately, because no one's really interested in indomethacin, there wasn't a lot of data on indomethacin as a repurposing drug. But what I've included is 3 plots against the dependency on the androgen receptor in prostate cancer. The first 2 are abiraterone, which is a very effective drug that indirectly targets the androgen receptor. Enzalutamide, again, a very effective licensed drug, which we're using clinically that targets this receptor as well. And then the indomethacin drug. And the thing to note is that this cell line DU145 is an androgen very well known to be androgen insensitive, and you can see no CRISPR dependency whatsoever, and very little activity from the drugs. And then these 2 cell lines, 22RB1 and LN-CLAP, which are known to be androgen sensitive, that are responsive to abiraterone and enzalutamide as well as indomethacin. And actually, there are scientists out there that are looking at the anti-tumor effect of indomethacin in castrate-resistant prostate cancer. Part of that's been attributed to AKR1C3 inhibition of that, but it may in fact be due to directly inhibiting the androgen receptor. So that would be, an ideal candidate for drug repurposing with this method. So I guess the conclusions of this this work is that in a real world setting that drug repurposing of currently untreatable, likely pathogenic genomic variants is feasible and may be possible in some patients with novel agents who would otherwise have no available treatment options, and that publicly available functional data suggests that results from drug repurposing databases may be functionally relevant. However, this may require further orthogonal validation. So I guess that leaves time for questions and discussion.
Thanks very much, Mal. Very interesting. I mean, it's really these sorts of issues at the absolute heart of this program, actually. So it's a very relevant topic, and it's absolutely true. I mean, we have there's so much sequencing data that we generate from patients that is kind of left over in the sense that once we filter out the initial series of mutations, or we have the kind of low-hanging fruit, if you like, but our ability or available methodologies to interrogate that kind of leftover data are very, very limited, and it's fascinating to consider that there might be methods to address that. So happy to take questions. Roger Daly.
Yeah, thanks a lot, Mal. Very, very interesting. And as Mark was saying, very, very kind of topical at the moment. So I noticed one of the first slides you put up when you were looking at the mutation spectrum across those patients indicated FGF19 amplification, and then that kind of didn't appear in the later analyses when you basically started to look for possible treatment approaches. And so I was just kind of wanted to query that, because I think there was like 9 cases of FGF19 amplification, and FGF19 is the ligand for FGFR4. And there's a couple of drugs in clinical trials for FGFR4 at the moment, which are in hepatocellular carcinoma, but it's also coming up in other cancers, including breast cancer as well. So I was just kind of intrigued by that. And I was just wondering whether your approach, if you've got a ligand, does your approach kind of just report on direct targeting of that ligand, or would it actually pick up the receptor for that ligand and whether there are drugs against that receptor?
Oh, you have to know it. We did this manually. I mean, we're trying to, definitely once you set through the ground truths of how you actually do it manually, you can actually program it.
Right.
But we've had to do it manually. So obviously I didn't know that was that. So I'll certainly add that to the analysis.
No, absolutely, because I think it's being used as a predictive biomarker for FGFR4 response. So yeah,
okay.
Yeah.
Thanks.
Okay.
Peter, follow-up.
FGFR4, yeah.
Thanks. It's very interesting. With the sensitivity data, I was just trying to get my head around that. You've expressed sensitivity as area under the curve.
Yeah, no, sorry. The curves are so. I have to relabel those graphs. So some of them are log fold change actually, rather than the area under the curve. So yeah.
So my question was twofold in a sense about both axes. I mean, one is it's purely, you talked about expression. I presume that's gene expression. Correct. Rather than the actual level of protein or the location of the protein.
Correct.
Which is an obvious limitation, sort of undergraduate limitation, but that is what it is. But the other question is, when you talk about sensitivity, is this all done at one concentration? Because clearly, particularly when you look at multi-kinase TKIs, concentration is absolutely essential. So
I'm going to do some work looking at the different dose levels. It's not. It's definitely not. They do have, for some of them, the dose level data, but they don't have it for all. So the reason why the axes are different is the GDS, for some of them it was a genomics of drug sensitivity screen, and for others it was a Broad Institute screen. So one used the AUC and the other used log fold change. So that's why and I forgot to change the labeling on the y-axis. Well picked up. The expression, the reason I chose expression was most of them were amplifications. So you'd assume that they would have high expression. That's the reason I chose it, but obviously it is highly limited. If anyone's got a better way of interrogating the data, I'm all ears, because this is something that I recognize as a limitation of these analyses. So if certainly if there are better or different ways to do it with sort of publicly available data, it would be tremendously useful. So dose levels definitely. I'm trying to figure out with the known biomarker combinations what some of the ground truths might be, interpreting that functional data, and then apply them to sort of new drugs.
Thanks.
Yeah.
Mal, is there are quite a few people around the world, and certainly within this group, that are interested in the concept of multi-omics profiling, so interrogating expression levels and changes in other molecules apart from DNA. Can you, I guess, postulate on how multi-omics profiling might complement this work, which is obviously just the most data that you've worked with here is obviously specifically focused just on the DNA molecule?
Yeah, I mean, theoretically, there's two separate things. So there's the general paradigmatic approach where you take a tumor type and then you take an off-the-shelf medication, right? So that, as a repurposing option. And then there's the actual method by which you choose how to repurpose it. So you could really theoretically substitute in any different method of, apart from probe minor, of how you're going to select the drug that you're going to repurpose. And you could use an appropriate tool for whichever platform that you want to use. So there's two separate things. You could apply that. Once you prove that this sort of paradigm or platform would work, you could certainly apply a different repurposing tool into that framework. It's a lot easier with genes than with proteins, and we just don't have, I don't think, the tools to do that yet. And I think that if you're going to work, with multi-omics data, a better approach might be to actually to try to do it sort of real-time functionally with live cells from the patient in some way, so either in vitro or with a rapid screening approach, functional approach.
And the final question is, what's your solution, or what are the emerging solutions in those patients that come through MTBs, not uncommonly, where there's essentially no low-hanging fruit, but you come out with multiple potentially functionally consequential mutations, sometimes affecting different pathways, and often making predictions as to what might be the key driver process is very challenging. Can you postulate on how we can use such tools to address those issues?
Automating it is going to be pretty difficult, but there's a lot of patterns. There are pretty consistent patterns that seem to affect certain pathways. So if you can sort of bucket them into multiple mutations within a certain sort of pathway, and there's a lot of pathway tools that are out there. You might be able to predict. If there were any machine learning experts out there, that certainly computationally predicting, which pathways are likely to be affected when you've got 5 genes, for instance, that are affected in that particular pathway. And then you can obviously use the allele frequency. So if there's a higher allele frequency, you might expect that that's more relevant, although that's questionable as well. And then, I think apart from that, I think we've got to try and learn. I think that's actually the best way.
Great. Okay, well, we're actually at end of time now. So thanks so much, Mel, for your presentation, and also for the attendees for dialing in today during what's a very busy pointy end of the grant season for us all. I'd just like to remind everyone that our next seminar is scheduled for the 27th of May. And our presenter on that day will be Professor Nick Huntington, head of the Cancer Immunotherapy Lab from the Monash BDI out in Clayton. So we look forward to that seminar. And please stay up to date with upcoming seminars and other events under the MPCCC umbrella via our website, monashpartners.org. Thanks everyone for your attendance. Thanks again, Mal, and look forward to seeing you at the next seminar.